This presentation is presented by Group 6.
Embryogenesis of the pituitary in the male and female reproductive tract. The embryogenesis of the pituitary in the male and female reproductive tract is a remarkably coordinated series of events involving the merging of several types of tissue that will ultimately form complete reproductive glands and organs. Here's an illustration for the derivation of the primary embryonic germ layers, wherein the inner cell mass or the ICM gives rise to the embryo and the germ layers, which are the endoderm, mesoderm, and ectoderm, that will develop into various specialized organs. During embryogenesis, various cells form organs that differentiate from discrete germ layers that make up the embryo. Differentiation is the process whereby a group of unspecialized cells develops into a functional, recognizable group of cells that have a common function. The germ layers that appear prior to embryo attachment to the uterus are called the endoderm, mesoderm, and ectoderm. These are the embryonic region of various organs and systems from the embryonic germ layers. Bold words indicate organs of reproductive importance. For the mesoderm, we have the muscle, blood vessels, reproductive system, which are the gonads in male and female, uterus, cervix, part of vagina, epididymis, ductus deferens, accessory sex glands, urinary system, and skeletal system. For the ectoderm, we have the skin, hair, nails, sweat glands, including mammary glands. For the nervous system, we have the hypothalamus, both lobes and pituitary, oral cavity, nasal cavity, and for the reproductive tract, we have the portions of the vagina and vestibule, penis, and clitoris. For the endoderm, we have the digestive system including liver and pancreas, respiratory system, and most glands. The pituitary gland originates from the brain and from tissue in the roof of the mouth. The posterior lobe of the pituitary originates from neural tissue of the brain. The anterior lobe of the pituitary originates from a completely different type of tissue in the roof of the embryo's mouth. Tissue in the roof of the mouth, called stomodial ectoderm, will give rise to the glandular tissue of the anterior lobe. Early in embryo development, a diverticulum, a sac or pouch diverting from a main tube, channel, or cavity, develops from the floor of the brain and grows ventrally toward the roof of the stomodium, the embryonic mouth. This diverticulum is called the infundibulum. At the same time that the infundibulum is developing, another diverticulum originates from the roof of the stomodium and grows dorsally. This diverticulum is called the Rath's pouch, or sometimes called Rath's pocket. As Rathke's pouch continues to develop, it loses its continuity with the stomodium and forms a discrete body of cells that become closely associated with the developing infundibulum. The cells of Rathke's pouch differentiate to form the adenohypophysis, or the anterior lobe. The prefix adeno refers to tissues that are glandular in nature, while the cells of the adenohypophysis are differentiating into various specialized cells capable of producing a variety of hormones. The infundibulum differentiates to form neurohypophysis, which is the posterior lobe. Embryonic development of the anterior and posterior lobes of the pituitary. The posterior lobe of the pituitary is formed from a diverticulum, which is a sac diverting from the main cavity, from the floor of the brain. The third ventricle is a cavity within the brain. Once this outpocketing begins to form, it is referred to as the infundibulum. The anterior lobe of the pituitary forms as the evagination from the oral cavity, referred to as the Rathke's pouch. The stalk of Rathke's pouch begins to regress and eventually completely separates from the stomodial ectoderm. At the same time, the infundibulum continues to evaginate to form the posterior lobe of the pituitary. Rathke's pouch completely separates from the stomodial ectoderm and becomes intimately associated with the new posterior lobe. The anterior and posterior lobes become surrounded by a deep recess of the sphenoid bone known as the cilia turcica, or the Turkish saddle. Migration of primordial germ cells from the yolk sac into the gonadal ridge. Primordial germ cells migrate by amoeboid motion around the midgut, enter the mesentery, and establish residence in the gonadal ridge. Migration of primordial germ cells from the yolk sac into the midgut region of the embryo as seen from the lateral view. The reproductive system develops in close proximity to and at the same time as the renal system. During its development, the embryo utilizes three morphologically distinct renal systems. The first is called the pronephros or the pronephric kidney. It is a non-functional remnant of a primitive form of kidney found in the lower animals. Early in embryogenesis, the pronephros regresses and is replaced by a functional bilateral pair of intermediate kidneys, which is known as the mesonephros or the mesonephric kidney. The mesonephros produces urine that is drained by a bilateral pair of ducts called the mesonephric ducts. Development and regression of the mesonephros with concurrent development of the gonad. Dorsal view. In the first illustration, we have here the mesonephros is closely associated with undifferentiated gonad. At this stage, the mesonephros is functional and is a simple version of the adult kidney. The mesonephros is drained by a series of mesonephric tubules that merge into a larger mesonephric duct that transports urine to the urogenital sinus. For the second illustration, the metanephros initially forms as a small bud from the caudal mesonephric duct. The mesonephros begins to lose its function and decreases in size as the metanephros decreases in size. The paramesonephric duct or the dashed line develops beside the mesonephric duct. Note that the gonad is also increasing in size relative to the mesonephros. For our third illustration, the gonad continues its enlargement, as does the metanephros. The metanephric duct will become the ureter. For our fourth illustration, the metanephros becomes fully functional and gonad becomes larger, while the mesonephros has almost completely regressed. In the male, some of the mesonephric tubules will form the efferent ducts, and the mesonephric duct will form the epididymis and the ductus deferens. The paramesonephric ducts degenerate in the male.
Sexual differentiation is regulated by a single substance directed by a gene on the Y chromosome SRY. The sex chromosomes within the spermatozoa determine whether the embryo will become a male and develop testes or whether it will become a female and develop ovaries. Females possess two X chromosomes, whereas males have one X and one Y sex chromosome. The Y chromosome contains a gene called sex-determining region on the Y chromosome SRY. The substance that controls the pathway toward either male or female development is the SRY protein, and it is controlled by the SRY gene on the Y chromosome. The X chromosome does not have such a gene. When SRY protein is synthesized by the sex cords within the primitive gonad, the development of the male reproductive system is stimulated. On the other hand, cells without the SRY gene express female genes and at the same time inhibit the male genes. It should be emphasized that development of female and male gonads is an active process in both sexes. In other words, the development of the ovary requires an active pathway but under female gene control. Therefore, ovarian development is not by default in the absence of SRY. The pathway of events controlled by SRY protein is presented in Figure 4 to 6. Figure 4 to 6. Pathway of events leading to formation of the male and female reproductive system. The sex-determining gene in the Y chromosome produces a protein that promotes the development of a testis. The primitive sex cords proliferate and penetrate into the medulla to form the testicular cords. Some of those cells differentiate into Sertoli cells, whilst the remainder become incorporated into seminiferous tubules. The latter are solid cylinders until after puberty, at which time they canalize. The testicular cords anastomose to form the ready testis, which becomes continuous with 15 to 20 persisting mesonephric tubules, the dactyli efferentes. The testis determining factor also induces differentiation of gonadal mesenchymal cells into the interstitial Leydig cells. For the female chromosomes, X chromosomes does not have SRY protein, and then the ovaries develop, and unlike in male, it has no AMH. Paramesonephric ducts become the oviducts, uterus, cervix, and part of the vagina, and finally, female tract is complete. Part of the male tract is derived from the mesonephros. Mesonephric tubules in the area of the gonad form the efferent ductus, whereas the mesonephric duct gives rise to the epididymis and the ductus deferens. In the male embryo, portions of the mesonephric kidney are appropriated for use in the reproductive tract at about the same time that the paramesonephric ducts begin to degenerate. Between 5 and 12 mesonephric tubules penetrate into the primitive gonad and make connections with the primitive sex cords via the ready testis. Ready testis is a network of tiny ducts that connect the seminiferous tubules to the efferent ducts. Efferent ducts are derived from the mesonephric tubules. The mesonephric duct will give rise to the epididymis and ductus deferens. Together, the efferent ducts, the epididymis, and the ductus deferens are appropriated to become excurrent extragonadal duct system of the male reproductive tract. Figure 4.7 Developmental sequence of the testis. First figure, a transverse section through the developing gonadal region of the embryo. The undifferentiated sex cords begin to align themselves with the small retitubules. The mesonephric tubules have not interconnected with the retitubules. The surface of the undifferentiated gonad is covered with a layer of connective tissue called the tunic albuginea. The paramesonephric duct or Müllerian duct is present but serves no function. For the second figure, the retitubules and the mesonephric tubules are now interconnected to provide continuity between the undifferentiated sex cords that are now developing into epithelial cords. These will become seminiferous tubules. The paramesonephric duct is beginning to degenerate. Degeneration of the paramesonephric duct is brought about by the production of anti-Müllerian hormone produced by the developing testis. For the third figure, the epithelial cords become seminiferous tubules. There is continuity between the ready testis and the efferent ducts. The mesonephric duct is gradually transformed into the epididymis and the ductus deferens. The paramesonephric duct has disappeared. The testes are formed at the level of the ribs. They descend into the scrotum late in gestation. In most mammals, the testes descend into the scrotum. Understanding testicular descent is challenging because it involves two relatively unrelated phenomena. First, the testis must travel a substantial distance from a retroperitoneal position in the body cavity to the scrotum. See figure 4 to 8. This movement involves a rapid growth followed by rapid regression of a ligamentous structure called the gubernaculum. The second challenge in understanding the process of testicular descent involves understanding how two layers of peritoneum cover the testis and descend with it. These two layers, the visceral vaginal tunic and the parietal vaginal tunic, are separated by a vaginal cavity that is continuous with the peritoneal cavity. The descent of testicles has three phases. They are: first, growth and elongation of the fetal body away from the testis; second, rapid growth of the extra-abdominal gubernaculum; and third, shrinkage of the gubernaculum within the scrotum. The testes lie in a retroperitoneal position and are attached caudally to the ligamentous gubernaculum. See figure 4 to 8. The gubernaculum extends caudally and resides in the area of the future scrotum. As the fetal body grows, the testes are pushed against the peritoneum. This pushing out causes the peritoneum to wrap around the gubernaculum and the testes. See figure 4 to 8. The first phase involves growth and elongation of the body away from the stationary testes. The second phase involves the rapid growth of the distal gubernaculum. The distal portion of the gubernaculum is the portion that has passed through the inguinal ring, see figure 4 to 8, and forms an outgrowth in the future scrotum. Two layers of peritoneum surround the descended testicle. After complete descent of the testes, you can see that the vaginal process is continuous with the peritoneal cavity and the testes is surrounded by a double layer of peritoneum. See figure 4 to 8. The layer of peritoneum immediately adjacent to the testes is the visceral vaginal tunic, and the layer away from the testes is referred to as the parietal vaginal tunic. The space between the tunics is the vaginal cavity. The space between the visceral and parietal vaginal tunic is continuous with the body cavity that houses the viscera. These tunicae are slippery and allow the testes to move freely within the scrotum during physical activity and during contraction of the external cremaster and the tunica dartos muscle. The most important component of testicular descent is the growth and regression of the gubernaculum. An important question is, what controls this growth and regression? The presence of the testes is required for the gubernacular growth to occur. Castration experiments indicate that the testis is essential for normal gubernacular growth. However, supplementation with gonadotropins and testosterone in castrated fetal pigs does not promote gubernacular growth. Therefore, it appears that there is a specific testicular components other than testosterone that causes the dramatic gubernacular growth associated with the first phase of testicular descent. Descent of the testicles requires both testosterone and insulin-like 3. Insulin 3 is a member of the insulin superfamily and is synthesized and secreted by islet cells in the pancreas and Leydig cells in the testes. Insulin 3 causes growth of the gubernaculum and testicular descent in male mice. Interestingly, overexpression of insulin 3 in the female results in abdominal descent of the ovaries. Figure 4 to 8. Major steps in the descent of the testes. Growth and subsequent retraction of the gubernaculum causes the testes to descend from the level of the 10th thoracic vertebra into the scrotum. Two common testicular descent abnormalities are cryptorchidism and inguinal herniation. Failure of the testes to descend into the scrotum is called cryptorchidism. The prefix crypt means hidden, concealed, or not visible to the naked eye. Orchid is a Latinized Greek word referring to the testes. Thus, the word cryptorchid literally means a testis that is hidden from view. Bilateral cryptorchidism results in sterility. However, cryptorchid testes are capable of producing testosterone. Thus, the cryptorchid male possesses secondary sex characteristics that are normal and has normal reproductive behavior. Inguinal herniation, on the other hand, is when a portion of the intestine passes through the inguinal canal into the vaginal cavity. Inguinal herniation is not uncommon in swine and occurs in about 1 in 200 males. Ligation of the parietal and visceral vaginal tunics is usually performed to prevent further herniation. Figure 4 to 9. Caudal view of a young boar with an inguinal hernia. The figure on the left shows the caudal view of the scrotum showing the anus, which is the A, the normal, which is the N, and the herniated, which is the H side. Notice that the herniated side is significantly larger than the normal side. The figure in the middle is an incised scrotum showing the normal and the herniated side containing the small intestines, SI, and testicle, which is T. The arrows indicate the parietal vaginal tunic, or PVT, covering all of the structures inside the vaginal cavity. On the right is a posterior ventral view showing completely exteriorized testicles, T, spermatic cord, SC, and small intestine, SI. The SI shown was previously within the confines of the vaginal cavity. The parietal vaginal tunic, or PVT, has been incised, thus allowing structures to be viewed. The point of entrance for the small intestine and spermatic cord into the inguinal canal, IC, can be observed. Figure 4 to 10, the position of the intestine after inguinal herniation using the bore as an example. Figure 4 to 10 is a sagittal view of the position of the small intestine in a bore with inguinal herniation. The small intestine sleeps between the parietal vaginal tunic and the visceral vaginal tunic.
Development of the female reproductive tract takes place in the absence of SRY protein by antagonizing the male pathway. In the absence of SRY protein, certain cells of the sex cords differentiate into primitive follicular cells and the bulk of the genital ridge becomes the ovary. Females have the X chromosome. It lacks the SRY gene that governs the production of SRY protein. As a result, cells in the primitive gonad of the female do not differentiate into Sertoli cells. Since there are no Sertoli cells, anti-Mullerian hormone cannot be secreted. Therefore, the Leydig cells cannot secrete testosterone and the male reproductive tract cannot develop. In the absence of SRY protein, the epithelial cords or the sex cords fragment into cellular clusters, each enclosing a primitive germ cell. These clusters of germ cells penetrate less deeply into the interior of the future ovary than in the male. Thus, primordial follicles are formed along the outer surface of the ovary that will eventually become the cortex of the ovary. Retitubule formation in the ovary is not pronounced, and a direct connection between the retitubules and the mesonephric tubules does not exist. Therefore, there is no tubular outlet for the gametes. The development of the follicles occurs throughout prenatal life and eventually the number of gametes will be maximized. Thus, the female embryo will be born with a pool of oocytes from which folliculogenesis will occur for her reproductive lifetime. Here are the major steps culminating in the formation of the ovary. In Figure 1, the paramesonephric duct and the mesonephric duct are still intact. The coelomic epithelium will develop into the ovarian cortex, and the remnants of the mesonephric tubules do not make contact with the retitubules. In Figure 2, the retitubules have disappeared and the paramesonephric duct continues to develop and enlarge. The sex cords begin to regress, but the primitive germ cells do not. After which, in Figure 3, primitive follicles or the black dots in the picture begin to develop at the periphery of the ovary. The mesonephric tubules and the ducts continue to regress while the paramesonephric duct continues to develop. Lastly, in Figure 4, distinct clusters of follicles surrounded by a single layer of cells develop at the periphery of the ovary. The sex cords have disappeared completely and the gonad resembles an ovary consisting of a cortical region or the region that contains the primordial oocytes and a non-germinal region or the medulla. The mesonephric tubules and ducts have completely regressed. The ducts of the female reproductive tract are provided by the paramesonephric ducts. The cranial part of each paramesonephric duct runs parallel to the mesonephric duct. The cranial part of the paramesonephric duct remains open to the peritoneal cavity, but the caudal end butts against the dorsal wall of the urogenital sinus and will fuse with it. The oviducts, uterus, cervix, and cranial vagina develop from the paramesonephric ducts. The paramesonephric ducts fuse together near their attachment to the caudal wall of the primitive urogenital sinus. The degree to which these ducts fuse determines the type of uterus the animal will have in adult life. Here shows the fusion of paramesonephric ducts with the urogenital sinus to form the vagina. The circled area in the picture shows the paramesonephric ducts that are beginning to fuse and will form the cervix and uterine body. A small bud evaginates from the caudal urogenital sinus and attaches to the fused paramesonephric ducts. Then, the paramesonephric ducts continue to fuse, forming a more defined cervix and a more developed vagina. The bud of the urogenital sinus also elongates. Lastly, the points of fusion have now formed a definitive cervix and a cranial and caudal vagina. The vestibule has stratified squamous epithelium because it originates from the urogenital sinus that invaginates from the exterior skin. It is important to note that both the male and female gonad and duct system originate behind the peritoneum, or retroperitoneal. Even though it appears that the reproductive tract, particularly that of the female, is inside the body cavity, it is indeed located outside the peritoneum. The supportive tissue for the female tract is known as the broad ligament and surrounds the uterus, supporting it from a dorsal and lateral aspect. In fact, the female tract is sandwiched between the genital fold. The genital fold will become the broad ligament consisting of the mesometrium, the mesosalpinx, and the mesovarium. Recall that the female tract is suspended by the broad ligament that is continuous with the dorsal peritoneum. This is the formation of the supportive structures of the female tract. It shows that the entire reproductive tract originates behind the peritoneum. In the more cranial region of the embryo, the gonad and the paramesonephric duct are quite separated. They may move ventrally as the arrow shows, but never entirely fuse. Thus, the ovaries and the more cranial portions of the future uterus and oviducts, or the paramesonephric ducts, never fuse in most species. In Figure 2, as the section becomes more caudal, the gonadal ridges as well as the paramesonephric ducts become more closely associated with their ventral movement. However, they still do not completely fuse. In the more caudal regions, the paramesonephric ducts have completely fused, thus creating either the body of the uterus, the cervix, or the cranial vagina. The remnants of the mesonephric ducts sometimes embed themselves in the wall of the vagina. These remnants can be seen in the adult animal and are called Gartner's ducts. It should be noted that the reproductive tract is sandwiched between two layers of peritoneum referred to as the genital fold. This connective tissue layer from the peritoneum forms the broad ligament that supports the female reproductive tract in the abdominal cavity. In cattle, a condition exists that results in abnormal embryogenesis of the female reproductive tract. This condition is referred to as free martinism. A free martin results from a unique condition during the formation of the placenta in the cow. In the bovine, the extraembryonic membranes of each conceptus fuse to form a common chorion. These membranes, or chorion, share the same cotyledons. Thus, there is a common blood supply between the male fetus and the female fetus. Because of this shared blood supply, each conceptus will be exposed to the same hormonal milieu. In other words, the female will be exposed to testosterone and anti-Mullerian hormone from the male fetus. Also, a free martin is a heifer born twin to a bull. A heifer calf is sterile, but the bull calf is fertile. In the bovine, the development of the testes occurs before the development of the ovaries. In fact, the testes are recognizable by the day 40, whereas the ovaries require several weeks longer to develop. As we now know, the testes secrete a substance called anti-Mullerian hormone. This hormone inhibits the growth of the paramesonephric ducts or the Mullerian ducts. Since the female twin is exposed to anti-Mullerian hormone as her reproductive tract is developing, the paramesonephric ducts do not develop completely. This incomplete development results in reproductive tracts that are blind, and canalization of the tract or the formation of a canal or lumen is not complete. In addition, the potential ovaries cease to grow and do not develop the appropriate complement of germ cells. Therefore, the ovaries are incapable of secreting estradiol and often secrete substantial amounts of testosterone as well as androstenedione. This atypical form of steroidogenesis not only causes abnormal female reproductive tract development but also programs the central nervous system so that the genetic female behaves similarly to a male. The response of the free marten to this elevated level of testosterone and androstenedione may range from the asymptomatic to significant bullish behavior. From a practical perspective, the free marten can be used effectively to detect estrus. Since these animals' central nervous system has been programmed to be male-like, they are generally more aggressive than a typical female in seeking out other females in estrus. By supplementing free marten heifers with exogenous androgens, maleness can be further enhanced. Further phenomena for fertility. All species do not develop a distinct sex or a separate testes and ovaries like this chapter explains. Some individuals possess both an ovary and a testis and are called hermaphrodites. C bases are synchronous hermaphrodites, meaning that fertile spermatozoa and oocytes in an ovotestes are present at the same time within a single fish. Self-fertilization is possible, but these fish have group spawning events to ensure genetic heterogeneity. Self-fertilization is advantageous if a C base is not present at the spawning event. In chickens and some ducks and doves, sex can be reversed after birth. Removal of the functional left ovary results in the development of the non-functional right gonad into a testis or ovotestis, or a gonad containing follicles of ovulatory size and tubules with spermatozoa. The younger the bird at ovariectomy, the greater the probability that the right gonad will develop into a testis. The older the bird, the greater the probability that an ovotestis will develop. Testicles sometimes stray from the normal path of descent. In humans, they have been found under the skin of the root of the penis and in front of the anus.